So the paper I'm going to be presenting is Circular and Longitudinal Muscle Shortening Indicates Sliding Patterns During Peristalsis and Transient Lower Esophageal Sphincter Relaxation by Nirali Patel et al. So some introduction material. The esophagus is part of the digestive tract with one purpose: to connect the mouth and the oral cavity to the rest of the digestive tract to the stomach. So material can either be put towards the stomach from the mouth downwards or to the mouth from the stomach backwards. There have been two major motor patterns in the esophagus that have been shown. Peristalsis, that transfers content towards the stomach from the mouth, so swallowing would be the best known example, and transient lower esophageal sphincter relaxation, also known as TLESR. In this case, contents go from the stomach towards the mouth. The best known example today of this would be acid reflux, in which acid from the stomach actually goes into the esophagus to cause discomfort. There are two layers of muscles in the esophagus: the inner circular muscle, also known as the CM, and outer longitudinal muscle, also known as LM. During peristalsis, it's been shown that the CM and LM contract together to move contents towards the stomach and towards the end of the esophagus. They relax together to receive the chewed up food in essence. In TLESR, only LM contraction at the end of the esophagus near the stomach occurs. This contraction then spreads towards the mouth, effectively bringing things up from the bottom part of the esophagus towards the oral cavity. A stark difference between TLESR and peristalsis is that there is no contraction of the CM in TLESR, while there is contraction of the CM in peristalsis. As you can see in the schematic shown here, the circular muscle is the inner layer of muscle within the esophagus, while the longitudinal muscle wraps around the circular muscle on the outside surface of the esophagus. The longitudinal muscle actually contracts to cause shortening of the esophagus, while the circular muscle actually contracts, causing an increase in the pressure. Through previous research that we've already discussed, it's been known that the two layers of muscle actually shorten differently relative to each other. This shortening that's actually independent of each other results in a relative sliding movement of the two layers of muscle to each other. So now that everything that we've discussed before, the purpose of the experiment was to actually document different patterns of contraction of CM and LM during TLESR and peristalsis. So now let's get to the materials and methods of the experiment itself. Even though the experiment had used both rats and humans for the specific study, due to time constraints, only the human study will be discussed. 18 subjects with an average of 32 plus or minus 12 years of age were used with no history of gastrointestinal surgery. All subjects fasted and had to stop smoking 6 hours before the experiment itself. The scientists placed a pressure-sensing catheter inside the esophagus. All subjects were anesthetized locally so as not to feel discomfort of the catheter inside their esophagus. The scientists made the subjects swallow water 5 times with 30-second intervals within each other, and the pressure was measured every time. They did this in 2 locations, 6 centimeters within the esophagus and then 12 centimeters within the esophagus itself. At both locations, the same procedure was used. Water was swallowed 5 times between 30-second intervals. The pressure readings then were obtained and were then converted into ratios of muscle thickness, which we will be observing from now on. So now let's move on to the results. For this figure B is a summary of the entire findings of the group in terms of the muscle thickness. SW stands for swallowing, which would be the peristalsis muscle contractions, while TLESR is the other type of muscle contraction in which contents would go backwards from the stomach to the mouth. What we see here is the maximum muscle thickness possible to the baseline muscle thickness ratio. The CM and LM again stand for circular muscle and longitudinal muscle. As we see in figure B, the ratio for peristalsis, swallowing, in the CM is very high and higher as opposed to the CM contraction in TLESR. In addition, we see that in the LM during peristalsis, the ratio is actually much smaller than for LM during TLESR. Finally, the third data set shows another ratio in which the LM and CM were compared to each other in relation to the max to baseline muscle thickness ratio. We again see a stark contrast in which the ratio is smaller in peristalsis as opposed to TLESR. The bars on the graph indicate standard error from the average mean. Based on these muscle thickness ratios, it's very clear that the CM and LM have very different muscle thicknesses in different types of muscle contraction, in this case swallowing and TLESR. This next figure is a schematic to show how the differences in the LM and CM actually look in relation to each other during each of the different muscle contractions, or lack thereof. The baseline panel on the left shows that during rest, the LM and CM are actually the same thickness with each other because the muscles are all at rest. However, the swallow panel, the peristalsis panel, in the middle shows that the LM is actually thinner and longer than the CM, and the CM is actually thicker and shorter in comparison to the LM. However, in comparison to the baseline, the LM and CM are both actually shorter and thicker in swallowing to varying degrees. Finally, in the TLESR panel on the right, it shows that the CM is actually thinner and longer than the LM is, which in this case is actually thicker and shorter. In comparison to the baseline on the left, it appears that the CM is actually quite the same, while the LM is shortened and thickened drastically. So, in conclusion, these results indicate that the two muscle layers that make up the esophagus actually move independently of each other, and that there are different types of shortening and thickening of the two muscle layers based on the two different types of muscle contractions. In essence, these muscle layers are not anchored on top of each other, and yet they slide over each other. So here's my works cited with the article itself cited and the image URLs. So now for some class discussion questions. The first question is, how might the longitudinal and circular muscles work to push food downwards in the esophagus into the stomach? So what I'm asking is, how do these two muscle layers that move independently of each other actually work together to accomplish the same task? The second question I'm asking is, why would it be detrimental for the two muscle layers to actually be tightly anchored to each other in terms of the function of the esophagus and the axis of the contracting fibrils? Remember, the axis of the contracting fibrils of the circular muscles are in a ring, while in the longitudinal muscles they're actually up and down in a line.